Good morning, everybody. Okay, so we are approaching this session. Should be very exciting. The promises and challenges of big data in neuroscience. Our first speaker will be Herwig Baier, and from the Max Planck Institute in Munich. And he will be speaking on neural and genetic architecture of visions. I hope you leave some time for discussions because that's the place where we can interact with you. Please.
So, thank you very much for the kind introduction. I'd like to thank the organizers for organizing this fantastic meeting and for inviting me. And it's an honor and a pleasure to be here. This is about the future of neuroscience. So I managed to sneak in at least the word vision into the title, although it's meant here in the sensory context. So our lab is using zebrafish to dissect behavior and the neural circuit underpinnings of behavior. Since this is a very diverse audience with varied interests, I decided to add a slide here in the introduction that explains our approach. We aim to identify the elementary components of behavior and then map them onto neural circuits. So you could call these elements, modules, motifs, atoms, syllables of behavior. We bridge molecular, cellular, connectivity, and network scales in our analysis. And we are also very much interested in the development and the evolution of neuronal connections. Also with a relatively new and recent interest in the distinction between machine intelligence and organismal or biological intelligence. Our institute, which many of you know, used to be called Max Planck Institute of Neurobiology, and now, starting January 1st, it's being renamed Max Planck Institute for Biological Intelligence. So we basically, this research is going to be extended, and we're going to transcend neurobiology and become more and more interested in ecology and ethology. So I'm using the words cognition, perception, intention here during the talk, and but I would like to warn you, those of you who are cognitive neuroscientists, that we pursue an unapologetically reductionist approach to these mechanisms in the brain, and I'll let you be the judge of how interesting and important and significant this kind of approach is. Okay, so zebrafish at the age when we study them are just a few millimeters in length. They're tiny, about the size of a fruit fly, but they're vertebrates, bona fide, have the same genes that we have. And one of their potentially most complex behaviors is prey capture. So here's the unwitting victim of this fierce predator. Can you? Okay. I move too much. Okay. And so what we are observing here is this zebrafish as it's sneaking up on its prey and it's gulping it down. And this is sort of the last snapshot in the life of this paramecium as it disappears in the mouth of the predator. And this is a beautiful behavior. We've done a very detailed kinematic analysis of the behavioral sequences that underlie this behavior. And for those of you who have been to the posters, there's beautiful work by Lilach Avitan here at the Hebrew University that refines this even more and brings in computational approaches to describing this behavior in great detail. I will not talk about this, so this is just to give you a flavor of the the goal-directed behaviors that these tiny vertebrates can exhibit. So in this talk, since this is about the future of neuroscience, I decided to basically walk you through an example of where we use our approach. And this is centered on retinal ganglion cells. So this will be a bit sort of a longer part of the talk. And also about the organization of downstream sensory motor pathways. And then I will give you kind of glimpses of flavors of how we want to extend this approach to cognitive processes. And then at the end, I hope I have time to talk about this, I'll tell you about a new pipeline which includes experimental and computational approaches where we hope we can accelerate the kind of discovery that we've used on retinal ganglion cells and speed them up by an order of magnitude and extend it to the rest of the brain. Okay, so retinal ganglion cells, as most of you know, sit in the inner retina, and they send axons into the central visual brain areas. I'll tell you more about this in a second. They receive inputs from interneurons here in the retina, which in turn receive information about photons hitting the back of the eye and are sensed by photoreceptors. And as this slide already indicates, these ganglion cells come in many different shapes. We've collected different morphologies of these cells, and they are small because the eye is also very small, but they assume these shapes that we know very well from other vertebrate organisms: mouse, chick, human, monkey, etc. So there's about 16 or so stratification patterns, conservatively estimated. And just for those of you who are not in the field, these different types of retinal ganglion cells are assumed to be feature detectors. So each of these types is tuned to detect a certain aspect of the visual scenery, such as on or off, motion in particular directions, chromatic content of the image. And this information is then bundled in the optic nerve and sent to the central brain. So these are the axons of the retinal ganglion cells as they enter the contralateral side of the zebrafish brain. And we and others have identified different target areas. So these are indicated here in these different colors, called arborization fields. They correspond to well-known neuroanatomically defined nuclei in the vertebrate brain. And most of these nuclei are conserved, so they are found also in mammals. The biggest one here on top is the optic tectum. So here's the tectum that receives the vast majority of retinal ganglion cell inputs. And it has a characteristic layered structure, which we have identified here by using rainbow labeling. So this is a stochastic labeling method that labels individual ganglion cells in different colors using a clever recombination strategy. And when we sort of look here from the top onto the tectum, we see this tangled mesh of differently colored axon arbors. And when we take a confocal image stack and turn it on its side, we see these 10 layers that each receive retinal ganglion cell inputs. Okay. So, the projector is not is not showing the yellows really well. So this is a schematic overview of, or a catalog of all the morphologies that we've identified. So this is now combining dendritic arborizations here. So this is the dendrite shapes in the retina with the axonal projection patterns into the brain. So all of this, you don't have to decipher this. These are is a list of the targets in the brain, and you can see that all of these different morphologies can be detected. Some very simple. Here's a bistratified ganglion cell that has just one layer in the tectum that it projects to, all the way to very complex ones with highly complicated collateralization patterns. So we have a full account. In fact, we have never, so this was a first survey of 500 ganglion cell type shapes, and we've not encountered an additional type since then, looking at thousands more. So a few years ago, Yvonne Kölsch in my lab teamed up with the labs of Josh Sains and Karthik Shekhar at Harvard. Karthik is now an assistant professor at UC Berkeley, and we transcriptionally profiled the zebrafish retinal ganglion cells. And this, so each of these dots is a cell, and this is a dimensionality reduced rendition of the transcriptional profiles of these cells. We can identify these clusters. There's more than 30 of these different clusters, and we can identify a whole list of marker genes that quite specifically, often just single type, in a single type fashion, label these subpopulation of retinal ganglion cells. Now, Yvonne set out to match the morphologies to the transcriptional profiles, and she generated with CRISPR-Cas9 genome engineering transgenic lines that label subsets. For instance, this is the MAFAA subpopulation of retinal ganglion cells, and she finds that this is a subpopulation that projects here into just a subset of the targets, 2 layers of the tectum, and one of these arborization fields, etc. So you can see now that the morphologies, the so-called M types, morphology types, have correspondences in the transcriptional types. And it's very interesting, and I think it needs to be explored further, that each type can be is specified by a very sparse combination of transcription factors, very similar to what's been shown in the spinal cord or has been shown in Drosophila or in C. elegans. So the morphology types, M types, find their equivalence in the transcriptional types. And we also looked at 2 time points, the larval retina and the adult retina, and find very interesting that there's actually dynamic remodeling going on. So many of the larval types, this for instance LC10, has an equivalent or has a progeny, which is the adult type. So there's often a 1-to-1 correspondence. There's also cases where a lineage is splitting. So these are post-mitotic cells that then assume 2 cell type identities in the adult. So 1 larval type, 2 adult types. There's also something really, there's a lot going on here in this tree. And for instance here, I've just indicated for 2 transcription factors how their combinations give rise to these different types. Okay, so going from the anatomy to the function, we can already state that there are parallel processing streams that extend from the retina into the brain, and their number is about 20. I'll show you. Just to give you an example of a dedicated processing channel, for instance, we found using functional imaging, not electrophysiology, but functional imaging, that there's one particular area in the brain, AF7, responds specifically to prey items. So when the fish is mounted in agarose and you show in a virtual arena a little dot that moves with the right kinetic parameters across the screen, the retinal ganglion cells project into this arborization field, light up. Other arborization fields are silent. There's also a layer in the tectum that responds very vigorously to these stimuli. So this AF7 responds to these small motile prey-like objects. It represents the frontal visual field. So it is served by retinal ganglion cells that sit in the temporal ventral retina. This is the location of the retina in which the image of the prey is formed as the fish is moving upwards to catch the prey. And it's also, and these retinal ganglion cells project into the anterior tectum. And this AF7, so they project here into the tectum. This AF7 area has a retinotopic map, but only of this frontal visual field. And using laser ablations and also optogenetic experiments, we could show that it is both necessary and sufficient to elicit prey capture. Okay, so these are the two types of retinal ganglion cells that project into AF7. You cannot see this due to the resolution of the projection, and they also form a collateral here in the optic tectum, in one layers of the optic tectum. And the marker that marks this type of population is MAF-AA, again, projection into AF7 into the superficial layer of the tectum. Okay, so, and this just, this shows the two dendritic types. There's a bistratified type, the diffuse type, and we think, we haven't completely shown this, that the combination of these two feature detectors creates to the fish an object representation that signifies prey. Okay, so in many cases we have now shown that the morphological types correspond to the transcriptional types and have a relationship, an equivalency with the functional types. Now, I didn't put equations here, but rather these equivalence signs, simply because often the relationship is complex. So the T types can be several M types, the M types can be several F types, so it's not a sort of a very clean one-to-one mapping, but these categories are overlapping in many cases, and I'll comment on that at the end of the talk if I have time. Okay, so to summarize a lot of work, we have assigned and annotated these individual pathways into the brain. So there is a system that detects prey, which I talked about. There's also a system here in the deeper tectum in which retinal ganglion cells project. So these are two layers that in the tectum that respond to looming. So these are usually threatening objects that extend in size, and the behavioral response to those is very dramatic. The fish make a fast escape from these looming objects. Could also be a collision, sort of a colliding object. There's also a system that responds to dimming, a very complex system. Often these dimming stimuli also elicit escapes. There's a phototaxis system here in the deeper parts. And then there is also non-image forming visual functions that are represented in these deeper parts of the visual projections. And also a direction selective pathway that is, we know this area is required for compensatory movements and also optokinetic responses that compensate for self-motion. Okay, so these are the retinal ganglion cells. They serve as feature detectors. The feature detectors are combined to give complex representations of stimulus, or they give configurations of stimuli. So how does the brain now take this information and processes it? Does this kind of labeled line organization extend into the central brain areas? And the answer is yes. So this is recording from 2,000 or so cells in the tectum. We can classify them by their response to, or cluster them by their responses to different visual stimuli. And there's a group of cells here that respond preferentially to small things that move. So small could mean distant. There is a group of cells that is not overlapping in the tectum that responds to looming or dimming stimuli. And then there is this large, about half of the cells that respond to moving dots or moving stimuli that are large, so in the range of 20 degrees or so. And these could, of course, be also things that are very close to the fish. So we found in work that I don't have time to show you that this system, it's about 2/3 of the tectum, is used to drive approach behaviors. And then there is also a substantial fraction of the tectal cells that are involved in avoidance or escape behaviors. So for the aficionados, I don't have much time to talk about this, but it's actually interesting to think reconstruct the transform function of how retinal ganglion cell inputs, which are depicted here, these are the stimuli that we used, are transformed into tectal responses. And what is striking is that the tectum response profile is very similar to the incoming inputs from the retinal ganglion cells. There is some additional processing, for instance, direction selectivity in one direction being added, but overall, the individual responses of the tectal neurons can be described or can be mathematically modeled by a linear combination of retinal ganglion cell inputs. Okay, so I told you about these two systems, the approach system and the avoidance system. What we discovered a few years ago, Thomas Helmrecht in the lab, a graduate student, discovered is that there are two parallel output pathways that originate in the retina. So these are different populations of projection neurons. One of these lines is dedicated to escape behaviors, and the other line is dedicated to approach behaviors. They have different targets, they have different collateralization patterns, and they also have different positions within this tract that leaves the tectum. So the medial tract that contains axons that, when optogenetically stimulated, induces an escape behavior, and the more lateral tracts, when stimulated, evoke an approach behavior. And there's, in fact, a topographic organization within these bundles, in that the axons that come from the anterior tectum tend to induce a forward swim, whereas the one that are more lateral here, the red ones, induce a greater tail deflection, which results in the fish making a larger turn. So there's a topographic organization here, and we call this a space code. So it's not a rate code, which would have been an alternative interpretation, really a space code that gives rise to these distinct motor lines for approach and escape, and even for gradations of the approach. Okay. Oh, it's not responding. I should tell it.
You could use the mouse.
No, but he also okay. That's the computer just restart. All right, now it's working probably. Okay, so I think it's probably froze for some reason. Okay, so I'm now going to talk about the approach system. I already showed you that there are dedicated motor outputs of the tectum, but one of the peculiar features of the approach pathway is the following. So if we think about prey capture, a typical hunting episode starts with an edible item appearing here in the peripheral visual field, perhaps at some distance. So the representation of such an object would be here in the posterior tectum, according to the retinotopic coordinates, and it would be small. As the fish then turns towards this prey item, the image, so to speak, of the prey moves along this tactile axis and will elicit or evoke activity here in the anterior tectum. And as the fish is now also approaching and bringing this prey item close before the strike, the representation will also be that of a bigger object. And we find exactly this, the distribution of these prey-selective cells, that the small object processing interneurons are enriched here in the posterior tectum, and the ones that respond to larger objects are enriched in the anterior tectum. And we carried out ablation studies to show that these cells have these distinct functions. So the retinotactile circuitry has evolved to accommodate the sensory ecology of the animal. And this is kind of a theme that we see often in the brain. The brain of these tiny vertebrates is not really interested in a veridical representation of the visual environment, but rather the circuitry was selected to serve the behavioral purposes of the animal. Okay, so how am I doing in terms of time? I think I have 20 minutes. 15 minutes? Okay, good. So I'd like to switch gears a little bit here and show you sort of a behavior that is more complex and involves social recognition. Because you may ask, well, catching prey for this little animal, this is just reflexes. So here we are looking at a behavior that involves an interaction with a conspecific. So, of course, zebrafish are showless, eager showless, and Ian Cousin will be talking about this behavior tomorrow in greater detail. Now, the first stages where we see a shoaling-like behavior is in juvenile zebrafish, 3 weeks of age. So these very tiny larvae, if anything, they avoid each other. The juveniles start to shoal, and they are without getting tired, they follow each other in a dish, forming these dyads. And Johannes Larsch in the lab showed that you could have fish respond to little dots that move beneath them. So this fish is following this little dot. This little dot is a representation of it's just a dot-like representation of this fish's position. So what we are watching here is two fish Skyping with each other, where by projecting the image of the right fish on below the left fish and vice versa. And when you overlay the two traces, it looks like these fish are actually interacting in a single dish. And Johannes could show that the attraction, the mutual attraction of these virtual avatars is as strong as it would be with a real fish, showing that this is largely visually driven. And of course, this makes it possible to do psychophysics. So now we can use a little dot that moves, that has different shapes, different sizes, different frequencies, different speeds. We can ask what are the optimal features to induce this shoaling behavior. And what Johannes showed is that by testing a large range of variables, he showed that it's the bout frequency with which this dot is jumping that it gives the information about whether or not this is an attractive shoaling cue. So he varied the bout frequency and measured the attraction, and this corresponds to, so the best frequency, the best tuning corresponds to the occurrence of the natural bouts in a real conspecific. Now, Johannes, a couple in the lab looked in the brain, doing whole brain imaging, and found, this is cutting a very long story short, in the dorsal thalamus, neurons that responded specifically to bouts. And again, the tuning very nicely matches the behavioral tuning. And we now reconstructed the circuit, identifying these putative bout preference neurons, reconstructed them using an EM volume, and determined sorry for the projection, doesn't show all the details of these neurons. Reconstructing all the inputs and outputs of these cells, and I'm going to go through this rather quickly because it's not as impressive on the screen as it is on my computer. We identified both the inputs to these cells, which come from the optic tectum. The tectum indeed is fed or receives these particular cells. They project into the thalamus, receive inputs from the retinal ganglion cells, and then the thalamic cells project into areas such as hypothalamus and preoptic region, which are known to be involved in social behavior. So this theme of labeled lines extend to these more complex behaviors, such as social recognition, and we believe it's a general organizing principle of the nervous system. Okay, so in the last, I'd like to sort of, because this is about the future of neuroscience, I'd like to give you a bit of a flavor of what we hope to do in the next few years and what we've already done towards this goal. And that is to look at elementary neurocognitive tasks that the fish brain can execute. So I took this from a review by Paul Sisek, who's attempted some kind of a taxonomy of cognitive processes here. And I think I've shown you that with larval zebrafish, we can pretty nicely cover the perception aspect and the action aspect. There's a lot of work that I could not talk about. So now the question is, can we also sort of go into this black box here and study cognition? So to define cognition, and this is up for a discussion, of course, one can define cognition as the internal manipulation of mental representations, right? So perception leads to a representation, which is basically an activity pattern. And once this representation becomes manipulated or some operations are executed on it, so giving a modified representation, we can talk about cognition. So the factors that can influence this manipulation could be on the short term or experience or anything that the fish has learned, attention, so stimulus selection, prediction, and they could also be longer term or long lasting things like what's the goal of the fish, what's the state, is the fish hungry or satiated, and also the history. So I'm going to skip this. I'll just show you one sort of glimpse of what we can do in the zebrafish, and that is looking at spatial attention. So in this experiment, we exposed a fish to a looming stimulus that was slightly displaced to it. So this forces, if we look at this panel here, this forces an escape away from the looming disk, right? So disk is presented on the left side, fish darts to the right, and vice versa. When the disk is on the right side, the fish moves to the left. This also works for weak stimuli, so these can be low contrast stimuli or slowly moving disks. It also elicits a very vigorous response away from the stimulus. Now when we show a high contrast stimulus and a low contrast stimulus simultaneously, the fish has to make a decision, and it recognizes with very great reliability the more salient stimulus and moves to the right, so towards the stimulus that would normally elicit an escape to the left. When we show two equally strong stimuli, the fish makes a decision. It either, and this is, and as we could show, is best described by a winner-take-all process. So there's a random selection of one of these stimuli. Fish either darts to the left or to the right. There's also in 20% of the cases, there's an interesting strategy where the fish swims through the middle. So we call this averaging. So we can study these processes of stimulus selection in the fish, and to cut a very, very long story short, we've identified circuitry in the tectum in this, and in one of its satellite nuclei, the nucleus isthmi, that can serve this kind of winner-take-all computation. And I don't have time to go into the details. This is all published. Now happy to talk with you about this during the coffee break. Okay, so a lot of what we've shown in this analysis is beautifully consistent with predictions of spatial attentional mechanisms. For instance, we find an inhibitory cell that has a very broad arborizations in the tectum that serves the global suppression of activity. We find a more focal excitatory cell that probably helps to enhance a local excitation. Okay. So to summarize this part, and I think I have 3 more minutes for, yep, okay. To summarize this part before I talk about the pipeline, what emerges are principles of visual system organizations that are somewhat different or have a different flavor to it than what you would read in the textbook. So stimulus features, as I showed you, are encoded by a large number of cell types. These serve as matched filters in engineering speak. Stimulus configurations are encoded in single neurons in the tectum by convergent projections of feature detectors. Efficient coding and the spatial arrangement of receptive fields serve behavioral utility, not a veridical representation of the sensory environment. Behavioral outputs are generated by parallel, segregated sensorimotor pathways, and I call them labeled lines. So it's much more similar to an insect nervous system than previously assumed. I didn't talk about these 2 things, but just I sort of touched on our work on spatial attention, which is implemented by local enhancement and global suppression or by averaging. And we have in the zebrafish pinpointed these mechanisms to particular subpopulations of neurons and their connectivity. Okay, so in the last minute, I'd like to introduce you to a computational pipeline that we think will speed up the discoveries of the functions that we're interested in and any other functions that we want to assign to parts of the brain. So I talked about T-types, which are the transcriptional profiles of individual cells and how they relate to morphology and connectivity, M-types, and how eventually we want to tie these 2 dimensions of the type to the function of the neurons. So we start with transcriptional profiling. This is work pioneered by Inba Scheiner, who is here at the audience. She, in this case, looked at the tectum and identified, very similar to what we've done in retinal ganglion cells, markers of subpopulations of tectal neurons. There's again the order of 50 to 100 different cell types. We can carry out in situ hybridizations. For this, we use a highly sensitive and specific new method called hybridization chain reaction, which can be multiplexed. It's fluorescent, so you can use different dyes and co-visualize the marker genes in the brain. So we know exactly where in the brain these markers are expressed. Can generate, of course, transgenic lines using genome engineering that give us genetic entry points into recording from these neurons and manipulating them, ablating them, or stimulating them. But most importantly, oh yeah, and I just wanted to show you this. This is not, doesn't look very impressive at these lighting conditions. This is the zebrafish brain. We've now reconstructed thousands of individual neurons. These little dots indicate the position of these neurons in the brain. And since we've reconstructed them, we can also investigate their projection patterns. So this is now shown in a few seconds. Yeah, so we can fill the volume of the brain now with morphologically reconstructed neurons. And all of this is summarized in our zebrafish brain atlas here, which represents different modalities. They're all in the same reference frame, regions, then neuronal morphologies, and markers, including lots of genetic markers. And with this, we can do systematic anatomical profiling of particular regions. And now the challenge has always been to then link the function to the anatomy. And for this, Johannes and Inbal teamed up to develop a pipeline. So we first do two-photon imaging of a brain. So here we're using a battery of visual stimuli. We save this fish in which we've done these recordings. We have thousands of recorded neurons in these brains. And we then fix the fish and subject it to multiple rounds of these in situ hybridizations with markers that we've identified. So this is now done in the same fish, in the same frame, in the same standard brain. You can anatomically register them and assign different genetic identities to the functional recorded neurons. And I don't have time to go into all of this, so I'm going to skip it in the interest of time. But the major gist of these kinds of analyses in the tectum is that we can really find, this is now the functional space, so we did a T-SNE clustering of the functional profiles of these cells, and then mapped onto these individual cells the expression of the marker gene. And we can find here little subclusters in these functional clusters that express a particular marker. So with this type of analysis, which we've done, as I showed you, quite comprehensively for retinal ganglion cells, we can now go into different areas of the brain and can, in principle, cell type any area that we're interested in the brain. And a 10-year project now becomes a half-a-year project. Okay, so I'd like to thank the people in the lab that have contributed to this work. This was at one of our recent retreats. I mentioned work by Johannes Kappel, here by Inbal Scheiner, and by Dominik Förster. And I'd like to thank you for your attention, and I'm happy to take questions.
We have 5 minutes for questions. Shalom, Moritz. If you can arrange the computer. Questions?
Very, very nice talk. I wonder how much there is learning and optimization after birth, and how much is basically programmed into the behavior, and the fish can do it with small fine-tuning? And do you think that basically what evolution is doing, like fine-tuning the circuit to have the behavior program, and it's very similar when you fine-tune the network while learning in the cortex, plasticity, but you do it because of learning in your lifetime, learning over evolution timescales.
So, by fine-tuning, by what? By learning? By experience?
I'll just forward it myself. I don't. Thank you.
So all the behaviors that I showed you are entirely innate. You can raise there's lots of interesting twists to that. So you can raise the fish in the dark, never seen a photon in his life, and expose him to, after a week, to prey, and he starts immediately hunting. Okay? Now he gets better. Also, we can show that if you raise them in a particular prey item, let's say in paramecia, and then give them a different prey item, such as rotifers, they have to adjust their behavior to that. So the kinematic modules are then shifted and adapt to the trajectories or the different kinetics of the prey. So there is a strong learning component. I would say the ground plan, the architecture is genetically determined, but then there's fine-tuning of the behavioral routines. We've raised fish that had no retinal ganglion cells. So this is, we use a mutation that removes all retinal ganglion cells. And then did transcriptional profiling of the brain, and we can show that all the cell types develop. They undergo the same differentiation trajectories, so express the same cascade of transcription factors, however a bit slower. So there is a contribution of the sensory surface, but maybe a bit disappointingly, it is largely in the secretion of a factor that advances their neurogenesis. But the connectivity appears unaffected, and the cell type composition is also unaffected. But there's a lot of, I could talk a lot more about this. Yeah, let's talk during the coffee.
You emphasized the labeled line picture. I wonder whether this is a product of the methodology or really the objective picture of the brain, because on the other hand, you talked about cognition as emphasizing the role of internal states and representations which integrate many influences which are counter, I think, to the picture of labeled line.
So we're moving. So this is exactly the tension in our work. I mean, there is evidence of cognition in the fish. I hope you follow me here. But then there are these rigid, hardwired pathways. So we know internal state, like stress or hunger, sleep. We know that there's a neural substrate for these, which is in turn hardwired. So it affects the processing of sensory information by modulating synaptic weights. And we've shown this a few times. For instance, hunger wakes up certain prey-selective neurons in the tectum. So there's a greater number of prey-selective cells in the tectum, and we know that this is a serotonin effect. So this is where the cognition lies in our view of the nervous system. But I think there's a very interesting tension. I'd love to talk more with really cognitive neuroscience people about how we can resolve that. So I told you I'm unapologetically reductionist, and so my view of the brain is that the brain uses repeating modules. So for instance, we know the elementary motion detector, but there may be similar modules such as a spatial attention module, which is then repeated in the brain, and the chaining of these modules then gives rise to cognitive processes, what appears to us to be mysterious processes where learning prediction plays into the function. So to be continued, and there's much more to talk about.
Absolutely, to be continued. Thank you very much again. Very pleased to have Moritz here. He developed a very beautiful department in Frankfurt on connectomics. Beautiful also aesthetically, but big and large and very active. There are several groups in the world that are doing connectomics. This is relatively new, I would say, field. It was not well appreciated 5 years ago, but now I don't think we can understand any system without knowing something about the connectivity at the level of synapses. So I'm very eager to hear you, Moritz.
Thank you so much. Can you hear me? Is this on? I turned it on. I think I turned it on. Can you hear me now? Yeah? Okay, it's working. Thank you. Thank you so much for having me here. It's a great honor and pleasure, and each time fills me with joy and gratitude that you invite me as a German scientist to Israel and to Jerusalem. Thank you very much, and that I can come to friends. Hi, Bidan, Ali. And I think the opening of this beautiful building, I won't forget a few years back, and now it's full of activity, and that's a great pleasure to see, and again, my great pleasure to be here. Now, talking about connectomics circuits, why are we so interested in this? One view that we have is, and it has already been touched upon in the previous discussion, there are things in this world that have been around for a very long time. Apples have been falling from trees, and bananas have been smelling or exuding substances, and birds have been flying for millions of years. And then there are, of course, other phenomena in this world that haven't been around for so long. And some of these are very beautiful, especially music, of course, that we have to learn individually. One kind of phenomena can be obtained and understood and structurally analyzed by evolutionary mechanisms, but others have to be individually learned. And it is definitely true for our brains, human brains, that we are very well able to do both. Some things must be evolutionarily prepared, other things, and that is a main part of what we experience as our ability to navigate the world, is individual learning, on which we base predictions, knowledge of the world. And now, interestingly, both of these converge structurally on the connectome. And here, I also mean to define precisely the synaptic resolution connectome, as you already stressed, Ethan. Because if you look at projections and tracts and so on, then you get some part of this, but not the individual learning. The individual learning, from all we know, goes into the synapses, so to say. So if we want to understand both contributions, and ideally quantitatively, and disentangle what's going on in various parts of the brains, I would argue we need the synaptic resolution connectome to structurally analyze them. And that is what we are trying to do. And then, of course, when it comes to actually using the brain, we use the connectomically stored information from these two main streams to take current sensory data and make sense of it. That's clear. But this connectomic part is what we are studying. And just to extend for one more minute on this, what this means, I think, on the evolutionary time scale, that this relative relation between, if you want, the predefined and the individual connectome may be dramatically changing. If we plot the state space size, so very simple thing, just the configuration space, yeah, that circuits can assume given the number of synapses you have and the number of neurons you have, then this state space, and this is double log 10 on the y-axis, it's dramatically increasing, of course, from simple worms, let's say, or even the stomatogastric ganglion, via flies, then the mammalian retina is in the middle, if you want, and then into the cortex, where this, of course, explodes. If you do the same calculation of state space for just the cell type connectivity, as shown here in blue, it's not as dramatically increasing, at least by current measures of what we call cell types. So I would like to emphasize this distinction between connectivity that is cell type based, and that is often with these ball and stick models that we use to understand parts of the brain, and then the cell type, then the cellular connectivity that is individual. Just to emphasize that just a little bit more, our first attempts at connectomics in the mammals was in the retina of mouse. That was actually the first mammalian connectome now almost 10 years back. And there, I think we still have an example of strongly evolutionarily defined circuitry, even in the mammal. When analyzing this with our techniques that I will introduce in a second, we came up with this cell type-based connectivity, where each entry here is a cell type, not an individual cell. And that allowed us, for instance, to explain certain features of certain types of ganglion cells, in this case, a well-described ganglion cell that Josh Sainz and Marcus Meissler had described that may detect predators. And this is important for the mouse. It makes sense that evolution took care of it, right? Eagles are very dangerous, and it makes a lot of sense to build precise circuitry in the retina of a mouse to make sure that each and every mouse will be able to evade these predators. And that is this kind of ball and stick circuitry, like in the bottom right, that we, of course, can obtain from dense connectomes. But that is, if you want, cell type connectivity. And a lot of connectivity we're analyzing these days, still in neuroscience, is cell type connectivity, again, sketched out down there. A connects to B, doesn't connect to C, and so on and so on. But what we are also very much interested in is if now cell type A doesn't have one or a few cells, but hundreds or thousands, like in the cortex of mammals, what could the structure of circuits in homotypic populations, if you want, be? Yeah? Not any more cell type connectivity, A connects to B, but among A. And just to emphasize this, this is where we think learning is happening, right? This is where we think storage of individual experience is happening, in the synapses between otherwise homotypic neurons. And by the way, this is also, I think, where the equivalence to AI lies currently. This is how AI, how networks, deep networks, RNNs are being trained right now, not cell type pre-structuring. pretty naive A-type cells, and then the weights is what's being changed. And both of that is happening in brains, and definitely in mammalian brains, and definitely in mammalian cortex. And this is really what we are very much interested in. So with this little bit elaborate justification, if you want, let's turn to what we're actually doing and how we're starting to make, I hope, progress on these fronts. So cortex, of course, is structurally a quite fascinating phenomenon with, in a cubic millimeter of mouse cortex, at least 100,000 neurons, kilometers of axons, a billion synapses, on that same scale also in human cortex. Now, that's dense and packed and so on, we know that. But I think the key structural phenomenon of cortex is that a given pyramidal cell, as the main cell type in cortex, connects to, as far as we know today, on the order of 1,000 other cells. That's not a precise number, it's an order of magnitude statement, could be a few thousand. But this is really the structural basis for a network that has at least external dimensionality on the scale of 1,000. I don't think there are other interaction networks that are so high dimensional, maybe social, but not really. Most people don't have 1,000 friends. And so this is really, I think, unique about cortical networks. And when we try to reconstruct these, it's quite challenging because we have to obviously follow all these beautiful cables that are also shown on the outside of this building, but not at their real size, which would be 50 to 100 nanometers in diameter for most axons of pyramidal cells in cortex. And as we know, of course, each of these axons is highly branched and extends over 3 dimensions. So we really need high-resolution, large-scale imaging techniques, 3-dimensional imaging techniques. And this is what the field of modern age, if you want, connectomics has tried to work on for the last about 20 years to get 3-dimensional EM, electron microscopy techniques that allow us to move forward. So what do? We take essentially a biopsy of brain tissue, and then we need to solve all the embedding problem, the chemical problem, if you want, of getting heavy metals into the things we want to see, not getting heavy metals into the things we don't want to see, so we have contrast, and then making everything stable by fixation methods and so on. So this is not easy, and actually my lab has contributed the 2 protocols that are used these days for large volumes, so the millimeter scale protocol and recently a whole mouse brain protocol. So this is really a must-have step that to do this kind of work. And then there are several techniques by now to automate imaging at the EM level. And the first one that came, so to say, or actually reinvigorated that field is the technique invented by Winfried Denk, the so-called block phase EM. I'll briefly show that in a second. But nowadays, we are mostly using this automated serial sectioning tape collection method developed by Ken Hayworth in Jeff Lichtman's lab. And there are other methods and even more. And by now, one has quite a choice of techniques for the various problems. Then there is some kind of automated image analysis. This has made enormous progress. We are profiting here immensely from AI developments. And then still a lot of manual proofreading that, however, we are now replacing by automated proofreading, so we get connectomes efficiently. One of these techniques, the serial block phase EM technique, works like that, just as an illustration. So we scan the tissue block that we obtained from the brain, and then with a scanning EM, and then have a diamond knife installed in the vacuum chamber of the EM to aberrate the top 25 or 30 nanometers, and then take the next image. And this is one way of automating this 3-dimensional image acquisition by sequential 2D image acquisition, of course, as in all other cases as well. Now, the data then looks like this. So this is now an example of such dense 3D EM data where you have everything labeled that you're interested in, and then, of course, you can start reconstructing. And any given cell is a very sparse subset of what's going on. Now, what have we been looking at? So the one system that we're very interested in as a model system, and many also here are very interested in, is the primary somatosensory cortex of mouse, so-called barrel cortex. This is a primary model system when it comes to the concept of so-called cortical columns, because it has been established decades ago that the main whiskers on the snout of the animals are via only 3 synapses matched to a network of about 1,000 neurons, 10,000 neurons in cortex, together creating such a cortical column. And this representation, as many of you know, is quite precise and resembles the representation of fingers, for instance, in other animals. So this is a very prototypic, if you want, sensory cortical column system. And in this system, we've started with looking at layer 4, where a large part of the thalamic input arrives in cortex. It was our first shot, and we really started with a very small volume there, but already there tried to ask the question, can we disentangle what is likely predefined circuitry at the cell type level versus aspects of the circuitry that could already be learned at the level of layer 4. Because we don't know where the storage of priors of sensory experience is in cortex, right? It could start at S1 level. Of course, we think it's especially in more frontal areas, but it could start at S1 level. So we wanted to look into that, took a really small but already quite dense volume of layer 4. Here you can see the cell bodies again. Blood vessels are these large white things here. And then, of course, all the fibers running up and down. The apical dendrites here, then you see myelinated fibers, the black and sheath structures, so to say, a high-speed transmission if you want. And then, of course, all the many synapses that are densely packed into such neuropil. The challenge, just to illustrate again, is to follow these axons here. Yeah? So these very thin structures, this was actually spine neck, but let's pick one of them here. So this is a thin axon making a synapse, then traveling on. Anyway, if you're used to it, you see it, but it's not trivial to follow all of that. So we started by reconstructing all the cells in this volume that together then already are quite a dense representation. But of course, the cells with their cell body is still only a subset of what you find in this volume. The main part are the axons. So 2 meters of axons in these about 100 micron cubic, 100 by 100 by 100 micron is already meters of axons. And then that together gave the first cortical connectome that we published a few years back, thousands of axons times thousands of dendrites. Now, we use that to do several analyses, again, defining inhibitory cell types and so on. But I want to share one particular analysis relating to the question, can we pull out what could be learned in such a connectome? Now, that's a bit counterintuitive at first because it's a static snapshot, so how can we talk about plasticity? But there is a trick that has been around in the field actually for a while, which is if you think about Hebbian learning, then, of course, first of all, we know that LTP, or Hebbian strengthening of synapses, leads to growth of synapses. That's lucky, so to say, for us. Synaptic weight has a physical, structural correlate. Larger synapses are stronger. That's a lucky kind of coincidence for us in our field. And then the trick is, if you look at paired synapses, then the prediction from Hebbian learning, of course, is because this electric activity in the pre- and postsynaptic cell are largely correlated on such small spatial scales. The prediction is if there's Hebbian learning, let's say LTP going on, it should be going on at both synapses together, right? And this concept that synapses should grow together if they are same axon, same dendrite, and undergoing Hebbian learning is something, again, that has been around already for a long time. Kristen Harris has used this before, and in particular in the collaboration with Terry Sinofsky, to pull out things they were interested in. We used this concept to ask, can we now pull out how much of the circuit is consistent with such a Hebbian learning? Again, I don't think I have to explain Hebbian learning at all here. We know what we're talking about, spike timing dependent plasticity. If a connection is used to activate the postsynaptic neuron, then it should be strengthened. And if you think about this, that if you have a pair of synapses that is both are undergoing LTP, then what you would predict here is that their strength is increasing, and thereby they are getting more similar in weight if you plot that here. So for some reason the movie here it is again. Yeah. So they're getting more similar in weight by getting larger, especially if they saturate. But even just arithmetically, even if they were not to saturate, right, they would relatively become more similar. So and that's all behind this analysis. That if you plot now instead the difference of synaptic pairs, the weight of synaptic pair difference with 0 being no difference, and then the CV here getting larger. And on the y-axis, the strength of the synaptic pair, so to say, so the weight of that synaptic pair. Then LTP, or Hebbian LTP, strictly predicts that you would move from anywhere in this plane up to strong and similar synaptic pairs. Yeah, that's a direct prediction of Hebbian LTP. And this is what we make use of, because now we don't have a handful, but thousands of synaptic pairs in our data. Yeah, 5,000 or so. And this is the distribution of synaptic pairs and their weight and their difference. So first thing you can see is that by no means is everything shaped and determined by finalized Hebbian state, right? Because these synapses will show up here, large and similar also. Yeah? They do exist, but there are also many, many synaptic pairs that are not cannot be the result of finalized Hebbian learning. Now, quantitatively, we can ask, how does the data we measure compare to a random shuffling of the same synaptic weights? and then take the difference as an indication of where potentially such Hebbian mechanisms have been at play. And in fact, we find over representation of large and similar synapses that quantitatively are around 20% of the synaptic base. And I mean, there are several caveats here. Obviously, if you have LTP finished and then you have diffusion of weights by homeostatic mechanisms, you move your synaptic weights away. If you have LTP first and then LTD, you will have a prediction of the trajectory in this kind of analysis plane. If synapses get eliminated, if synapses get made new, you have predictions on this data. And this is my point here. This analysis is still very simple in the sense that I'm using the most simple picture of LTP here, but the more principled approach of taking a learning model that you may like or have, and now it has to suddenly fulfill or match these constraints here. So we have very strong constraints now on a circuit and the distribution of synaptic weights in a circuit, especially via the paired configurations, that any learning model that you like should be able to reproduce. Right? So in that sense, I think it's really a novel kind of constraint that we can use at scale. Yeah, I don't have to illustrate that here in this audience that knows a lot about these things. So some of these pairs are consistent with saturated LTP. And obviously we can then roll this out for certain types of connections, especially we're interested in the thalamocortical connections because these were described decades ago already to be using NMDA-dependent plasticity in a critical period in the first postnatal week to form these barrel maps. Yeah? So now we can say that only about 10 to 15% of the connections at 4 weeks of age, so adolescent, are still possibly the result of this process. Yeah? That we can say for sure, because if you have Hebbian LTP and it's stable and is maintained, then we should be seeing that. And so in that sense, we can drive bounds on the part of the circuit that can have undergone these processes. Okay, so this was a first analysis that already started or tried to relate to quantitatively assessing how much individual learning can be in such a cortical circuit. Obviously, early days and first steps, so don't get me wrong, I'm not claiming we are there, but I want to emphasize this possibility to test learning models on massive structural data like we've just illustrated. Now, the second story I want to tell relates to the more probably genomic encoding of circuitry and the development of that in the individual. So we asked, can we use our techniques to now screen across postnatal development in a given species, how these circuits are put together. And for this, we again used barrel cortex of mouse, but now early days. So those first synapses are made around P5, intracortical chemical synapses around P3, 5 on that scale. And then we took data sets at these various time points. So here's this first postnatal week that is a critical period in barrel cortex, and then at P14, things become a bit more mature, but P28 is what we would call adolescent, at least in mouse. So we took many data sets. This is the heroic work of a PhD student, Anjali Gaur, who took most of these data sets, so not one, but 13 or so in the end, trying to sample these relevant time points in postnatal development. So if you look at the cortical structure, then at P7 things are still very sparse in the sense that you see hardly any synapses, hard to find them. P9 gets already a bit better, more easy to see synapses, more mitochondria as well, by the way. P14 then already almost like what we know from the adult cortex, and then at P28 things look like we've seen that before. Now, obviously, the fact that synaptic density increases is nothing new at all. This has been established for a long time, that you have obviously an overall even overshoot probably of total synapses, but also GABAergic synapses. So this was not the point. The point is, we know that these GABAergic synapses undergo or have a lot of specificity with respect to who they target. Right? This is this whole beautiful world of inhibitory axons going for the soma, one thinks PV, basket cells, and so on, going for apical dendrites, distal dendrites, somatostatin, and so on, or going for other interneurons, or axon initial segment, Chandelier cells. So these are these prototypical inhibitory targeting properties, and we asked how do they develop under this, if you want, curve of overall synaptic density increase, but how are these specificities established? On what time scale quantitatively? And this is what we analyzed. We started with the somatic innervation, so the innervation of cell bodies by inhibitory axons, and quantitatively use the simple fraction of target choice, so to say. So if you have an axon, you have synapses, then you can simply ask how many of the synapses are on the right target, so to say. Yeah? So this is a very simple measure of specificity, and then that can increase. Now, again, for somata, for apical dendrites and axon initial segments. If we start with the apical dendrites, the distal dendrite innervation, then the axons that innervate distal dendrites, again, think somatostatin and so on, are already very specific at the earliest time points we measured, even at P5, 30% preference already for apical dendrites. So there's almost no time course. This was not significant here. So it's really the 20 to 30% preference for the relevant targets is already there essentially at birth, or we cannot say at birth, but when the first synapses are formed, it's already there. And this is in stark contrast to what is happening with soma innervation, which is almost absent at P5. So, but then within a few days, rises up to about 20% specificity. Now, this is quite a dramatic change, as I will try to illustrate it. It's also quite different, again, from what we see with the apical dendrites. So somatic innervation is essentially absent early on and then rises dramatically within just a few days during a time when apical dendrites, distal dendrites are already very well innervated. So how would you do that? How can you increase synaptic preference for cell bodies? Well, one option that one probably first thinks about is to add synapses that go onto the right target, right? You start with, let's say, 10% preference, and then you add specific synapses. That's actually not so easy to do because the additional synapses now have to be very specific, right? Because all of them essentially now have to be on the right target. But there's an alternative way, and that is, of course, synaptic pruning of the wrong targets. Yeah, if you take away the synapses that go on the wrong targets, you can also enhance specificity. But if you compare these two models, they make a very interesting prediction, which is if you add synapses that are on the right target, your synapse density will, if anything, increase a bit. But pruning, in order to really go by a factor in specificity of 2 or 3, as we've seen in the developmental data, means also reducing synapse density by a factor of 2 or 3. So dramatically, measurably dramatically. So this is something we should be able to measure if that's happening. So we wanted to compare these two models and ask what's happening with these soma-specific axons. And now when we actually plot the synapse density, we in fact see from 7 to 9 exactly when the specificity increases. We also have a more than twofold decrease in synaptic density. So here the population shifts from high synapse density, low specificity to lower density, high specificity, quantitatively again summarized here. Apical dendrite innervating axons don't really change their density, but soma innervating axons reduce their density also by more than twofold over the time period where they increase their specificity by 2 to 3-fold. So visually, it's also very clear. Yeah? So at P7, you have these many black synapses, so to say, loaded with synapses off target, and then 2 days later already they are gone. Many of them are gone, and it's becoming specific. So that means we found that for these subtypes of interneurons, we have differential likely mechanisms of postnatal development and establishment of target specificity, pruning as the most likely mechanism for somatic targeting over just a few postnatal days, which actually extends to the perisomatic synapses. So not just those directly on the soma, also proximal dendrites, they are preserved, the others are taken away. Now, what about AIS innervation, chandelier cells, very briefly? These don't occur so much in layer 4, so we went to layer 2-3. And this, again, is a prototypic specificity example, right? So the axon initial segment of pyramidal cells gets innervated by inhibitory synapses later. Yeah, at P9, this is essentially absent. It's happening 2 weeks, 4 weeks. That's known that it's a later development. What was not so clear before is that, in fact, a large fraction of the innervation of the axon initial segments is not made by these specific chandelier axons, but by the off-target innervation by, let's say, soma innervating axons. So actually half of the synapses are not by chandelier cells. But let's focus on those chandelier cells, so to say, and then we can see that over time, the specificity of these chandelier axons here shown in magenta increases. Again, quantitatively at P9, there's a little bit already, but almost not distinguishable from unspecific innervation. But then here's now the AIS innervation plotted. At P14, they are roughly half of their synapses are already made on axon initial segments, and then by P28, they are very specific, but even more specific at several months of age, where essentially all of their synapses are made only onto axon initial segments. But then all the time there's also this population of axon initial segment innervating axons that are not specific for doing that. Yeah, that is important to keep in mind. Now, for the chandelier axons, it's very interesting because, for the axon initial segment, they are all inhibitory, the synapses. Exactly. So it doesn't go back to another axon initial segment like the chandelier would, but goes to a soma or so. It's still inhibitory, but doesn't care about axon initial segments specifically. And that's really, quantitatively a relevant source of input to axon initial segments that I think so far has been overlooked. Now, for the chandelier development, and that's the last point about this individual development, which also I thought was quite interesting. We know that in the adult, the prototypic formation of these chandelier axons is the so-called axonal cartridge, beautifully described by early anatomists. These are these synaptic, bombardments along the axon initial segment, right? Cartridges, they are called. And it was thought for the longest time that the development of the specificity is tightly linked to these cartridges. Makes sense, right? That you would co-align with the axon initial segment and then make your synapses on these axon initial segments. What we find, however, is that already at P14, where we have none of these cartridges yet, there is already specificity for axon initial segments. And now we saw that this specificity is already coming back to the same axon initial segment. So in other words, the chandelier axon that later will form cartridges already at P14, where it doesn't form any cartridges, is able to find AIS and systematically so. So here, for instance, an example on different branches making 3 already synapses on the same AIS, not just on any, on the very same AIS. And again, if you think about how little axon initial segment there is in a given cortical circuit, just a stretch of 50 micron per cell, and all the rest is everything else, dendrites, axons, everything else, but not AIS, it's really clear that this is a highly specific process and here clearly not requiring the cartridge formation. So this, I think, is another insight from that study. So to summarize the postnatal development, we found that there are differential mechanisms very likely at play for soma innervation, distal dendrite innervation, and AIS innervation, and that's all published. So I will now jump to the final part. We then wondered, what about evolution? Because obviously that's the other big time scale that we all are interested in. In particular, when we talk about cortex, we want to know, of course, how much is the local cortex of a mouse comparable to the local cortical circuit of human. And I think so far we are still embarrassingly, let's say, uninformed about this. I mean, it's not so clear what really the main differences are. There are some beautiful results about capacitance, for instance, that Idan has worked on and collaborators. Synaptic measurements, I think there's also clear that synapses are quite strong. But when we really come to the circuit, so what is different at the circuit level? And this was the question we asked. Obviously, this is a long time scale, 100 million years roughly, our last common ancestor with mouse. We also took in macaque as comparison, so to be sure that we are not, overfitting on some human data that comes from the surgery room. So we wanted to be sure that this is a general principle that we're looking at. We took data sets across this time scale. If you look at dense EM, again, it's embarrassingly unclear. I put the labels here, but by now I think I would know what to look for and probably find the differences. But here it is now a bit over contrasted. But still, it's not super clear what the difference is. Now, when you look at cortex as a whole, so these are now all of the millimeter from pia to white matter in mouse and all of the about 3 millimeters from pia to white matter in human. Yes, it's 3 times thicker. What else? Now, the first thing that jumped at us was this. If you take mouse and simply label the most simple thing you can do, which is excitatory neurons in magenta, inhibitory neurons in white here, and if you now do the same in human, it's very clear that what is the most massive change is in about 3-fold increase in the relative fraction of interneurons in human compared to mouse. Now, very recently, there was transcriptomic data coming out of the Allen Institute also pointing that out, but that was really structurally something we were very much astounded by to see that so clearly. So about 3 times more interneurons, relatively speaking, compared to excited neurons in in human. Now, what does that mean? That, of course, immediately triggered our thinking about in human, inhibitory, excitatory, balance questions, of course, that I almost don't dare to bring up here. But of course, if you have one interneuron, so to say, relatively speaking, and suddenly you have 3 times as many, would that mean that then you have 3 times more inhibition relative to excitation on the postsynaptic targets? Dramatically, not by 10%, by 3-fold shifting the IE balance. Is that what is going on in human? Well, obviously, that would have dramatic consequences, not only because, and again, with time here, I'm almost not daring to bring this up, but of course it would be quite a dramatic quantitative change about the stability of the networks. And then also, it is still one of the key thought models, if you want, in many neuropsychiatric disorders to think about EI balance to change, but on the 10, 20, 30% level. What if now in human it's already 3 times off from mouse? So we needed to understand that. For this, we had to really carefully map the synapses on the distal dendrites, especially the basals, especially of pyramidal neurons in mouse and human, to understand what's going on. And we saw that it's actually more complicated. So what's happening is, first of all, and really very clearly, spine rates drop from mouse to macaque in human, 2 to 3-fold. Therefore, yes, it looks like you have relatively more shaft, which is mostly inhibitory, in mouse at least. So it did look, yes, like there is a 3-fold increase in, almost 3-fold increase in relative inhibition. So that was the first thing we saw. And we thought, okay, maybe that's happening. But then when we looked more carefully at the axons, we realized there's something else going on, which is that the axons of pyramidal neurons in mouse are always very much preferring spines, and interneurons only innervate shafts. That picture also changes in human. Excitatory axons make many more shaft synapses. And we first didn't understand how this comes about, but it's primarily, we think, an effect of distance from the cell body. So something that we had found before in other circuits. So in the dense population, we also see that shift, that the spine preference is much stronger overall in the dense axon reconstruction in mouse than in macaque and human. And the key effect, we think, comes from this distance-dependent synapse positioning, where proximal to the soma, you first innervate interneurons and use shaft synapses, and later you turn to spine innervation. This is something that we had found in MEC a couple of years back in medial and rhinal cortex, and something that had also been observed in bird before. But it's really very strong in human as well. So it's really a very strong sorting principle that we also find in human, where the synapses are first made onto shafts, and then later onto spines of excitatory neurons. Now, taking all of that together, and I won't bother you with the details, it's also published, we essentially find that EI balance is not changed 3-fold. Yeah? Essentially, in macaque human, EI balance measured here as I inhibitory inputs by all inputs is still around 10 to 15% or so. So it's not dramatically changed. Raising the question, where does all the inhibition go? Right? If you have 3 times more interneurons, they are not increasing the I onto E. What is going on? And that was very interesting because we saw that the interneuron pool expansion is not commensurate, so to say. So you have, even if you take the most simple distinction of multipolar versus bipolar interneurons from mouse to human, it's the composition changes also dramatically again by more than 2-fold the contribution of bipolar interneurons. So bipolar interneurons make up about 20% of interneurons in mouse, meaning 80% are multipolar basket cell-like interneurons, very clear. In human, it's only 50-50. So again, a dramatic change on top of the 3-fold overall change, a dramatic change of the subpopulation composition. And these bipolar interneurons make more synapses onto other interneurons in human than in mouse. So we have more of them, and they make more II synapses. So really the story is mouse to human, the main local circuit effect is 3 times more interneurons that make 10 times, in human actually 10, macaque is 9, but 10 times more II synapses, relatively speaking. So we think the key evolutionary effect from mouse to human is at the local circuit level, when you talk about these circuit components, is an II enhancement, not i.e. II, summarized here, tenfold expansion of the interneuron network. Have to watch the time. How much, do I have more minutes, or? I think 36, well, no, I think I timed it, 36 minutes. We started later, 36 minutes, thank you. Then let me briefly go one step further. We also wondered about the total input to pyramidal cells, because that's another topic that is in the textbooks, actually, that a human has so much more inputs, spines, but because we saw so many less spines, we quantified this in larger data sets and saw that, in fact, the total input, at least for layer 2-3 pyramidal cells, is not changing multifold from mouse to human. So really the only thing that strongly changes at the circuit level is the interneuron network expansion. What could this mean? Well, one thing could be timescales. A recent paper by Terry's group, carried this out, but I think it's already in the earlier work, that essentially if you have more II, you can get longer timescales. So they, made this very explicit in this study here for monkey cortex, that they can expand the working memory timescales even by just, 30% change of II strength. With a tenfold change, I'm not sure about how it extrapolates, of course, but it's you can think about it. If you essentially inhibit the inhibition, that you prolong the timescales. But again, just at the network level, it's very clear that with tenfold more JII, so to say, here going back to Haim's early work, that it's a different. It still can be stable, of course, but it's a different world. And then I think that the other thing that this raises for the future is, could it be, if now I had, if I have a network component that I don't really have in mouse, could something go wrong? And then could that create pathologies that we cannot, in this case, study in mouse? So with that, I would just like to give you one outlook for scale. Obviously, we want more than just local pieces of cortex. And this is really just briefly showing where we are now on the unpublished side. So taking an entire barrel, but ideally actually going for an entire column using new technology. This is the multi-beam electron microscope developed by Zeiss, which scans in parallel 60 or by now 90 beams. And with that, we are able to acquire data sets of that scale. So the entire cortical thickness of mouse in S1 here that then contains multiple cortical columns in this data set, and we've now been able to analyze this data set using our automated methods, so that we now have this, these are about 50,000 cell bodies, and then we define the cortical column based on the thalamic input to layer 4, about 10,000 neurons, and these we have now all reconstructed. This is now this cortical column reconstruction on the postsynaptic side, if you want, but we have also now achieved the presynaptic side reconstruction. So this is not a drawing, this is the real thing. You can't see anything if you see it. Yeah, obviously we have to downsample, add the spines automatically, and then we get here just a few examples of these beautiful layer 4 spiny stellates, and then using automated 3D tracking networks, we were able to essentially complete these axons to a point where we get reasonable recall and very high precision. This is a trick or a method developed by Martin, who really 3D flies through this tissue, very much like autonomous driving, but in 3D. And with that, we then have a barrel reconstruction and actually a whole column reconstruction at much faster than anything before, and the first cortical column connectome. And I leave it for next time to talk about what we find in there. But that's the scale at which we can now operate. Okay, with that, let me close and summarize and also emphasize the perspectives. Obviously, this notion of understanding invariances in cortical circuits with the aim of maybe getting at main processing streams, but then also imprinting or memory that is individual storage, if you want. And then, of course, the question, could things go wrong? So we are working on these 3 axes, time scale of the individual, of evolution, and then I haven't talked about that yet, obviously also the search for pathological change, which requires screening, and that we have also massively expanded, so now being able to screen hundreds of samples, that is to come. So the future, I think, is to take structural data seriously. It's not explaining everything by no means, but I think it will be the structural constraint that is very serious and very helpful, hopefully, also for many theoretical approaches, I think, because it has to fulfill these constraints in the future, and maybe putting that together can be fruitful. With that, I thank you very much for your attention.
Questions?
Thank you very much. That's stunning work. I would have 3 short questions. One is, feel free to answer any one of them or none. The first is, in the portion on Hebbian learning, you show different trajectories, but it seemed, some connections stay stable, some increase over time, but it seemed that around P5 and 9, there was always a dip, and I was wondering whether that's a measurement error or whether that's something else. That was the first. The second, you talked about individual brains, different from evolution, but do you also already have data or something say about really 2 individual brains differing, and how would you go about that? And the third part is, for your last science study, you talked about the difference between mouse and human and how much that changes, and that's very important for, let's say, neuropharmacology. But from vision and electrophysiology, the difference between macaque and human might be more interesting, and I would like to know how big the differences there are. Thank you.
Okay, briefly, so the postnatal changes, I think that you referred to, the P5 and 9, so yeah, I mean, there is an overall dramatic synapse density change, of course, yeah, and so everything we've now pulled out is under the hood, if you want, or under the envelope of dramatic increase in synaptic density overall. So that is one thing that is happening at the same time and may have these effects. Not everything, I mean, any major statistically significant effect we have tried to understand, some of these are also just not large enough in magnitude that we didn't care about the power to distinguish these things. So we picked these main effects, multifold changes. So that's that. Then briefly the last point, because I remember, I fully agree. I mean, obviously, an additional, I mean, I would maintain that mouse-human is important to establish, or mouse-primate as well. Now, macaque-human is also very interesting. We have not seen, at the level we've looked at the circuit level, we have not seen, paradigmatic changes, but quantitative changes. So the network expansion, the II network expansion is, in fact, even from macaque to human, another factor of something. 1.5 or so. So that is one aspect. The other is, and that I didn't include today, there seems to be also quite a difference in this whole Hebbian learning thing. But I have, so that's really very different, where macaque is like mouse and human seems very different, but that's something to come. I'm not yet sure enough, otherwise I would share it, but I think there's something that I'm not yet 100% sure it's true.
Can you ask from there?
In mouse. Yeah. Well, so far only mouse, macaque, and human. But we have rat, but that might be like mouse, and then we have some other species. But in terms of the expansion of that network, I fully agree. It would be very interesting. That's a question I would have also to you, whether it would be clear how that, because if you both have the supragranular expansion to start with, that you have described beautifully, and then on top of that, the fractional increase of interneurons, you actually need an even bigger factor of quantity, like of numeric increase of the interneuron population, and whether that's already clear how that is happening. But you already said that it's potentially just the thickness of the amount of CGE available. Just a comment: we are not yet sure whether the VIP interneurons, it's just the same interneurons. What we do see already is that they're even more specific in macaque and human than they are in mouse. So we find no single interneuron in mouse that only innervates interneurons. Even the VIP, 60, 70%, not more than that preference. But in macaque human, it goes up to 100%. So that is on top of every, and it could be that it's really new subcell types or new cell types that would still have to be described. So in that sense, yes, it is VIP-ish, but I would be ready for more change at the circuit level, from my point of view.
Okay, let's take a break because we have 15 minutes and then we reconvene here for the next lecture. Thank you very much, Moritz.
Yes, of course. Thank you.
לשבת לך אנחנו להחלקה. אוקיי, וזה גם לשים לה את המצלמה לראות אותנו? זה נחמד, בסוף לפחות, שאלות. אוקיי, טוב, בסדר. נשים את אני במצלמה, שייסה. טוב, אני אטפל בזה. It's different. Look, you can see that when you connect with computers, you have a very large screen and
okay, so but I can use mine.
It's up to you. Yes. You're welcome. Test. Test. Test.
Hi, Daniel.
Hi, can you hear me?
Yes, yeah, I can hear you.
Perfect. They will be a little bit late. You have the schedule already, yes?
Yes, no worries. I just thought I'd connect early just in case.
Yeah, they went out for a break, so they will
No
probably
problem.
be late
Yeah, that's good.
5 minutes. I'm trying now to bring a camera so you can see the people inside the hall. I will try to make some changes because the microphone is very sensitive. I will try to make it less sensitive. Yes, can you hear me now better or the same? Yeah, can you hear me now better? You are on mute. Can you hear me? Oh my God. Microsoft Can you hear us now better?
Yeah, sounds great.
Okay, perfect.
Looks good too. Nice auditorium.
You missed it.
I know. I wish I was there.
Okay, so we will be back when all the people are inside.
אין לך פה? אני רוצה להראות את זה לעידן. לא, אני רוצה להראות לו את הלינק.
Mackenzie, can you try to share a screen?
Yes, I can. 1 second. I just played up my slideshow here. Okay, what are you? How does that look?
That's great.
Okay,
Perfect.
excellent.
So you can just keep it as is and just mute yourself, or you can also you remove the camera until you see people coming inside the hall.
Perfect, sounds good, thanks.
You're welcome. Bye for now. Mackenzie? Yeah, the people start coming in, so I just want to, yes, exactly. That's perfect.
Great. Thank you.
Mackenzie, can you hear me? Yeah? Good. Maybe
Yeah, definitely.
yeah, maybe we can make you a little bit bigger on the screen. Tiny bit. When it will be projected?
As you like. I'm happy to
yeah,
be
we control
small.
it from here. Yeah,
That's
okay.
great.
So everyone is coming in now, and maybe 2 minutes until everyone is.
Yeah, no problem. Yeah, thanks for the great view. I can see the audience, which is a.
You can see the audience. Great.
Yeah, it's great. Yeah.
Okay. Fantastic. Okay. Okay, so we're a little bit late. Dan has to go to teach, so I'll step in. Unfortunately, Mackenzie Mathis had to cancel in the last minute, so to come in person. So we'll project her lecture. And today, I can't see the title, or it's somewhere here.
No problem.
So Mackenzie Mathis from EPFL will talk about learning, latent embeddings of behavioral and neural data. Please.
Yes. Yeah, thank you. Thank you so much. And first of all, I'm very sorry to not be there in person. As a Californian, I always have to adapt to the snow. And unfortunately, I took a little bit of a fall this weekend, so travel wasn't an easy option. But I'm delighted to virtually be here. So thank you, Haim, and the other organizers for having me. And so today I'd like to tell you about some new work coming out of my group. That was done with a PhD student, Stefan Schneider, and a master's student, Jin Lee, and myself. And I think many of you in the room here today share the sense of that one of the biggest quests in neuroscience is this neural origin of behavior. And we try to link underlying neural dynamics to the actions of the animal via neural computations. So in our laboratory, we're particularly interested in neural computations during adaptable and flexible behavior. So namely, we want to understand how they learn and adapt their behavior on very short timescales. So this requires us to have a program that really is broadly reaching. So in my group, we combine neuroscience, neuroengineering, machine learning, and computer vision, things like Deep Lab Coach I'm not going to talk about today. But altogether, what we're trying to do is combine these discrete fields into something where we can allow us to search for neural computations to actually build more adaptive AI. So as you all know, the field is going a remarkable technology revolution. Now we're able to record from thousands of neurons simultaneously. This is a great graph from Cording and Stevenson that was recently updated, just showing you the power of Moore's law, if you will. But really, we're in this regime where we can record from even a whole brain of an animal like a zebrafish, which I'm sure was already discussed today. I'm sorry to have missed it, or C. elegans, and even a significant portion of the mouse cortex with things like light bead microscope, up to 1 million neurons simultaneously, and certainly with neural pixels that give us much better temporal resolution. Thousands of neurons can be recorded at the same time. On the other hand, we and others have developed computer vision tools that now allow experimentalists to begin to capture behavior with even greater detail than we previously could. So anything from 3D kinematics to centipedes, where you certainly wouldn't want to label this by hand, to social animals like marmosets. And I think this collectively is showing us that we're in this era where we have these really large neural data sets on one hand and potentially very large data sets of animal behavior on the other hand. And so we and others have found ourselves in this situation where we can do, say, something like this in my laboratory, where we have mice that play with joysticks and we see how they adapt, and we can simultaneously record their neural activity. And I think this combination of large-scale behavior and neural data is really calling for new methods, particularly nonlinear methods, to link the brain and behavior at the population level. And so that's what I'll mostly focus on today. And I think this was really nicely laid out ahead of time by Anne Churchland and colleagues last year, where they published this very timely review in Nature Neuroscience, sort of a call to action, if you will, highlighting that no longer are these technological constraints keeping us to recording one neuron at a time. And so it really behooves us to start considering neural population dynamics more collectively at the population level. So in this work, they highlighted that things like decision-making variables or movement can be found nearly all over the dorsal cortex of mice. So how do we make sense of this? Mark Churchland and colleagues have shown that there's signatures of neural computations at the population level that don't show up in any one individual neuron. So I think that's a really important point, that there might be more to this than at the individual neuron level. But at the same time, many of the behaviorally relevant signals have been constrained to relatively low dimensional manifolds. That might be because our behaviors are too simple, or it might be because the neural code is truly just very low dimensional. I think at the same time, many of us are used to looking at plots like this, where you have neurons over time and they're ranked by their peak activity. And so you kind of get this snake-like plot. But we should remember that even randomly connected RNNs can give us such structure. So I think it really makes a lot of sense for us not just to consider individual neurons, but actually what might be the underlying causal latents that drive such activity. And so sort of to conceptualize this, what I want to do is just give you this kind of toy diagram and remind us that at the end of the day, what we're really trying to understand are the hidden underlying factors in the brain. But we can only observe a small amount of neural data. Even though we're at this great intersection where we have lots more neural data than ever before, we can still only record from thousands of neurons, not billions of individual neurons like in the human brain or millions of neurons in the mouse. And so ultimately what we're trying to understand with this neural data is what are the true underlying latents. And so where we sit in the field is that we actually want to learn these mapping functions to say, can we identify what the true underlying latents of this neural code might be that is actually observable to us? And so one of the ways that we and others, of course, have gone about this, or I'll tell you about our way today, but how others have gone about this, of course, is very smart with dimensionality reduction. As I mentioned, that a lot of the neural population dynamics are laying on this low dimensional manifold. And so the general idea is that if you have all these neurons, it's still a relatively high dimensional space, and you'd like to learn the structure in a lower dimensional embedding way, right? So these are tools like TSNE or UMAP, or DMix PCA or PCA or variational autoencoders, or some newer generation tools like PI-VAE or LFADS or GFPA have been used to do this in the neural code. And so for us, we wanted to think about this in two regards, which I'll break down through this talk. One is that how could we actually leverage behavior in finding these neural latent spaces, and can we do this in a hypothesis-driven manner? And secondly, we really wanted tools that were nonlinear and didn't have strong assumptions on the underlying data process. And I'll also break this down. But to get started, what we did is we took a synthetic data set that was introduced in the PIVE paper. And the general idea here is, of course, we want to discover these robust underlying latent dynamics and measure the statistical structure of these neural populations. And so synthetic data allows us to test this in a very principled way. So this might be familiar to many of you, but just in case, each one of these points would be the population vector at a particular time. This is a population of 100 synthetic firing neurons, and we've just given a behavior 0 to 2 pi in this data set, so it's colored by this roughly in the sine wave. And so ultimately, there's many tools that do a part of the job that we wanted to do, but nothing that we felt could do all of the things. And so I'll tell you what the laundry list was that we wanted from an algorithm to do this. So first of all, we wanted this to be really highly performant. We wanted latents that were interpretable, and one of the downstream tasks in that sense could be decodable. But more importantly to us is that we wanted consistency in the embeddings. That means if you run the algorithm 10 times or 100 times, you should get the same embedding space, not just cherry pick the one that looks the prettiest and put it in your figure. And we also wanted to find consistent latents across animals doing similar tasks. So if we believe there's a true underlying latent that is underwriting a behavior that is obviously manifested from the neural code, we should be able to find consistent latents across animals doing similar tasks, even if it's not identical. We also wanted tools that are identifiable. So identifiability is closely linked to consistency if you consider this from a nonlinear ICA problem. And what it means in terms of identifiability is that you can find the true model parameters, or they can be recovered up to some transformation here, often linear or affine. We also didn't want any data type assumptions. So we actually didn't want to use a generative model. That's because we didn't want to have users or ourselves change the underlying data generating process if we wanted to use, say, 2-photon calcium imaging, neural pixels spiking data, EMG, pure behavior, etc. Ultimately, we wanted to be able to use this behavioral information, but in two important ways. One would be for real-world continuous data, right? Behaviors are often not discrete, but then also still using it in the discrete setting. Lastly, we wanted to be able to use it for hypothesis, or i.e. supervised or discovery-driven unsupervised analysis. So the same tool should allow us to explore both spaces. And I think importantly for this session is that we wanted to be able to combine data from across animals, right? We can only see so much of the neural code at any one time, so we need to be able to build algorithms that can take inputs from multiple animals, even potentially using different data modalities. And so over the last few years, what we've been working on is an algorithm which we call Zebra. So it's an algorithm that allows for joint modeling of these auxiliary variables, i.e. potentially these behavioral labels that are discrete or continuous, and any time series data. So I'll walk you through this diagram here to give you a conceptual framework for how this model works. So the idea is that you have some time series data. We'll consider neural data today. This could be, say, calcium or spikes. And then we have these auxiliary variables that can come in two forms. One is just time. Ultimately, what I'll tell you, this is a self-supervised algorithm. So if there's the only, I guess, major assumption, if you will, is that there's something that's changing in the latent dynamics that you're interested in. So time could be an interesting way to discover these changes in the latents. The other is more supervised, i.e. you can truly use the behavioral data to, stripe, hence the zebra, mark or use this as a fiduciary marker in your neural data to actually then use this in the contrastive loss, which I'll tell you about in just a moment. So the idea is that you build up this tensor of positive examples. So when the behavior happened with the neural data, right, we're going to train the algorithm on the neural data. These are just the auxiliary variables, as long as negative samples and then reference samples. So it's a very classic triplet loss scenario. Then you have some nonlinear encoder. The structure of this actually doesn't matter very much. This could be a CNN. That's what I'll use for most of my talk today, but it could also be an RNN or transformer architectures. The important thing, and I think our contribution to this work, is that we modified the standard info NCE loss to allow for different sampling schemes, namely in the continuous space, which hadn't been done. I'm not going to get into the theory today, but there's a preprint that's underlying this. And for you that are more mathematically inclined, there's the proofs in the supplementary notes that you can check out for showing why this is identifiable. But importantly, for today, the idea is that this is just an encoding model, and then you can take this final layer output and build a low dimensional embedding in this way. So as I mentioned, we started with this artificial neuron data set because this allows us access to the ground truth or ground truth latents because we generated them. And this is the reconstruction that you get with Zebra. And we compared this to a class of variational autoencoders and also UMAP and TSNE. And there's two features I think that are important to point out. So one is that Zebra, first of all, has the highest reconstruction score of being able to actually recover the ground truth latents. So this is just an R2 linear regression between the ground truth and the reconstructed, allowing for transforms in all of the algorithms, or there's linear transforms in all of the algorithms. The other is this Poisson VAE, which presented at NeurIPS. And you can see it has a very classic behavior from variational autoencoders, right? They can get stuck in local minima. But for all these algorithms, I'm showing you all 100 runs. So occasionally it can be very good, but on average, it's not. And of course, in real world scenarios, you would never know what the ground truth underlying latents are. So it's very hard to know what regime you're in with this model. So in that regard, I think variability across runs is also a really critical feature that we wanted to optimize for. You can see UMAP is actually quite good at this, better than TSNE. But of course, this is on this synthetic data set, so your mileage may vary. But in short, I'm not going to, given the time constraints today, I'm not going to go into great detail of the mathematics, but what I will show you is that under very minimal assumptions, Zebra can recover these ground truth latents, and this is backed by a theoretical guarantee. And we show this in the identifiability proofs. And so I think Zebra is a potentially interesting model for data sets that you want to be able to leverage behavioral information, or of course, just using it in time series data. So concretely, I think neural data is an interesting use case for this, but so is things like even RNA sequencing data across developmental time points. Things like this type of time series data could also be used in this algorithm. But I'd like to give you some examples now of where we've tested it and how it performs. So let me introduce you to a data set that was collected in the Bujaki lab, where there's rats that are running on a linear track. It's about 1.6 meters. They're recording from CA1 in the hippocampus. And as probably many of you know, these include place cells. So there's 120 neurons recorded simultaneously in this animal. And let's just imagine we have a hypothesis that, the hippocampus maps space, which I think is pretty good given the Nobel Prize. But nonetheless, I think this is a good data set to introduce the algorithm to you. So as I mentioned, what you can do is build up these positive and negative pairs. So your sampling scheme matters a lot. And in this way, you also want to maybe not pick discrete points, but also these continuous distributions around particular positive and negative pairs. So if we had a hypothesis that the animal's hippocampus neurons are encoding place, then you could build a hypothesis like this. Or of course, you could start potentially by just doing this in a purely self-supervised fashion. And so we embed these on an in-sphere, so you're always going to see them on a hypersphere. You can also do fast ICA on top if you want to show it in a Euclidean embedding space. We also have an MSE loss that allows you to do this not on this hypersphere, but for many of the theoretical guarantees, this is a really nice space to optimize on for contrastive learning, as some of you might know. Okay, so the first thing is I'm just going to show you the discovery embedding. So this is only using time information. So this is fully self-supervised. There's no other auxiliary variables. And we've just post-hoc colored this by location on the track. So hopefully what you can appreciate is that, again, each one of these points is the neural code at a particular space and time. And you can see that the position is quite nicely encoded in the neural data, right? We can see the track on the left versus the right runs of this animal. And then we can use a hypothesis, namely, can we find a positional encoding even more strongly in this space? And so with this in-sphere, I'm also showing you the first three latents always, but these are 3D models. But of course, you can make n-dimensional models, as we'll get to in a moment. But importantly, hopefully what you can see is that now I'd say it's even a crisper space. Zebra is not a visualization tool, but at the end of the day, I think it's also nice to visualize your data. And so here we can see that there's this both potentially directionality and position mapping in these way. Now what we can do is keep the neural data fully intact, but just make an erroneous hypothesis. So just scatter these positive and negatives across the track. to see if we actually find random place, it would be kind of random place, if you will, coding. And what will happen is that because this is a really hard optimization problem, then the algorithm won't converge, and so will just collapse. So I think this is a feature, not a bug, but mainly what this means is that you can rule out hypotheses that are just really not in your neural data. So I think from a practical standpoint, what people might do is that you'll probably use the time algorithm first, look for structure in your data, and then try to understand the underlying latents that are actually driving this structure in your embedding space. So if you have a set of hypotheses, you can find those that might be more correct or those that are incorrect. Zebra is very fast to run and algorithms. You can set up a very simple sweep, and obviously these are too small to read probably, but the general idea is you can find features that are encoded in the data and other features that are likely not to be encoded in the data. But beyond the visualization, this is really backed by numbers. And so there is a goodness of fit metric with the info NCE loss. So you can say which hypothesis is most or the best fit to your data. So truly a goodness of fit measure. And so if you look at these loss curves at a particular point in time, an equivalent point in time, you can really see that, of course, this shuffled behavior, even though the neural data is intact, isn't converging. And you can see that position and direction is a nice hypothesis or a fit to your data. And as I mentioned, this goodness of fit is theoretically defined. So this number actually has a meaning, but I'm not going to get into that today. Okay, but one of the things that I kind of harped on in the beginning is that I'm really interested in consistency of these algorithms. So this is all on one rat, but you can train this, of course, on the 4 rats of the data set that are presented in this paper, and we can look at how consistent they are between rats. So here on the left, you see zebra behavior. This is just a 3-dimensional model. You can get higher numbers if you want with higher latents, but we'll get to that in a moment. But what you hopefully can appreciate is that if you treat one rat as a source and the other rat as a sink, you can see quite high consistency. You're finding the same latents in these 4 different animals, both in zebra behavior, also in time, to some degree less. But this is in pretty stark contrast to these other algorithms. So PyVAE, as it was implemented in NeurIPS, didn't allow time. So to be super fair, we included a variation of PyVAE, which we call conv-PyVAE. So using the exact same backbone as in Zebra, and just showing that if you use test time labels, you can still get decent structure. But if you don't use the test time labels, which are often not available, you don't get this structure. And this is also the same for TSNE and UMAP on this rat. But also the dimensionality of this space becomes super interesting. So another thing that we wanted to look at, now that I hopefully have convinced you that Zebra might be an interesting tool for doing hypothesis-driven neural population dynamics, is that we wanted to look at the topology of these spaces. So for that, first I'll just show you a schematic of how persistent cohomology is computed. So each data point is thickened to a ball of a gradually expanding radius. So then you can track the birth and death of the cycles in each dimension. I won't get into all the details, but in short, you can get these barcodes and say, what does the space actually look like via the Betty numbers? So for example, if you had a ring-like structure, you would get Betty numbers of, the H0 through 2 here of 1, 1, 0. If you had 2 rings, you would get 1, 2, 0. And nicely, I think, as I mentioned with the shuffling or these erroneous hypotheses, given that they collapse onto the sphere, you get this sort of 101 topology. And so what this allows us to do is to say what how do I put this? What dimension is the neural code, or how long does that hold up across the space until it starts to fall apart? So ideally what you can find is that a particular space that this might actually start to decrease. And so, for example, in the hippocampus data, you find the same persistent topology structure up to 16 dimensions in this rat hippocampus data set. What each one of these dimensions are actually relating to is still an open question, but you can go back on and map these onto the co-cycles from the coordinates. So it's really beautiful represented in the neural code in this way. But beyond topology, which I would also point you to these excellent papers, especially the Chowdhury one, which really nicely highlights why topology is an interesting feature for looking at neural population dynamics, and then the recent Gardner paper from the Mosers as well, looking at torus topology in grid cells. But one of the things that we wanted to do with these spaces, as I mentioned, is also be able to use them for downstream tasks like decoding or classification. And so with that, what I'll show you is this 3D embedding space with the 3 latent dimensions. You can then look at the time series dynamics of each one of these embeddings. Here are all the neurons just plotted. Here's the raw behavior of the rat running left or right, and then the decoding on this space. So this is a very simple decoder, right? Because Zebra is basically an encoding model. We just put a simple KNN decoder on the end, and it shows, I think, pretty good performance. Compared to, say, other algorithms like PyVAE or t-SNE or UMAP or PCA, which is often used, especially in the motor field, for doing decoding, Zebra does, I think, a quite acceptable job in this way. So we hope that this is an algorithm that allows you to look for consistent spaces across animals, but also can then be interpretable in downstream tasks like decoding. Okay, but this is a relatively simple task, right? This simple behavior, we're treating the rat as a point mass. And so we also wanted to look at other behaviors, like in the motor system where my lab is more firmly anchored. So for that, we turned to an open source data set from Lee Miller's group in Chicago, where they had monkeys making this center out reaching movement. So this is like a top-down view of them holding a robotic manipulandum. And in half the trials, or in a larger fraction of the trials actually, they're making active movements where they're cued to reach to a particular target. And then occasionally they're bumped. So the joystick just moves their arm. And this is actually encoding in S1 primary somatosensory cortex of these monkeys. And so first I'll just give you the qualitative results. So if you look at Zebra versus PyVAE or t-SNE or UMAP, hopefully you can appreciate that the structure is really reminiscent of the actual behavior. And we can also do decoding with really better performance than this data set has been used for decoding tasks before. So it shows state-of-the-art decoding on this task, but also shows you something about the dimensionality of the space that's needed. So maybe unsurprisingly, but sort of interestingly, you only really need 8 dimensions to actually decode quite well, both the active or the passive trials. But I think for today, what I really wanted to show you was this data set that I'm pretty excited about. So this is the Allen Institute has collected 2 different data sets where these mice, as part of a larger series of stimuli, are watching this so-called natural movie 1. These are different mice, of course, that are being recorded from. So they're either being recorded with neural pixels probes, or they're doing 2-photon calcium imaging, primarily in layer 2-3, but this is GCAM6 imaging as well. And so there's a few papers that have presented this data and then also looked at the sort of representational drift, which is a really nice paper in biology as well. And so what we wanted to ask in the context of Zebra is to say, first of all, can we use sensory stimuli as behavioral labels or these auxiliary variables? And can we actually find similar latent spaces across 2P calcium imaging and neural pixels data? I think, at least for me, this is a very big question, or still an open question, about how consistent or relatable these latent spaces are to each other in this way. So to do this, what we did is we actually took the movie frames from this natural movie 1. We ran it through a pre-trained Dino model, which is a state-of-the-art sort of representation learning space that does really nice segmentation of feature objects, as you maybe can see in this tiny video here from Meta AI that was just published at iClear. And then we can look at sort of the representations that come out of Dino and use these as the feature set to actually train Zebra with. So this is just over time. You can see 4 different transformer types, and you get pretty similar representations. So we took these features as the labels, if you will. Right. So just to remind you, what this would look like is, can be any behavior. So you can use many different features, like 500 different features or less or more as you like, in this way with the neural data. And then we built up this tensor, and we trained the network with our contrastive loss, as I mentioned, and you get these low dimensional embeddings. So we asked a series of questions. So 1, can we find evidence for movie feature representations in the visual cortex of mice using Zebra behavior or time? Could we produce similar embeddings from either neural pixels or this GCAM6 calcium imaging data? Can we actually, given that we have no generative model and no constraints like it has to be Poisson or Gaussian, can we leverage this cross dataset input to train Zebra, like a Zebra model with both inputs to do? And then ultimately, how interpretable are these? So could we decode the movie from the neural latent embeddings? So there's many mice in the data, but I'll just show you 4, just to give you an appreciation of that. First of all, in this neural latent space, we're training this with a higher dimensional model, but I'm just plotting the first 3, because that's what we can show very easily, is that you see this nice representation of time in all 4 of these animals in the behavior. And if we run this with time, so we're not using any of the neural features, we actually see when the mice watch 10 repeats of this movie, you actually see this, this feature set, this, of course, post-hoc color-coded now, right? You see this feature quite nicely delineated across these 4 different mice, showing us that we think there's something, you know, real in this latent space across these animals, even with this time one, if you're more skeptical of behavior. Importantly, again, we can do the shuffle just to show you that these again collapse. So we don't think that Dino is forcing this structure, because of course, if we just shuffle this, you'd still see the structure in the Dino features as well. But this is all trained on individual mice, but we can train models together. And so I'll talk about the decoding in a moment, but hopefully what you can see now with 4 different mice, you can train them individually and you can also train them together. So if we then post-hoc plot each mouse separately, you can see really highly consistent embedding spaces across these 4 animals. And importantly, as I'll show you downstream, the decoding is interesting. I'll talk about what that is. But just to convince you here that even though you can make highly consistent embedding spaces in this multi-training, so going up through time is later in training, we still tap out about the same decoding performance, even if you train individual mice, suggesting that we're not losing something fundamental to aid in the decoding performance of these animals. But getting to my first question, the first point was, could we actually find topologically similar embeddings across these different models? So what you see here are neural pixels only trained models with only 10 neurons. This also gives you a sense of how much neural data you need. So by about 100, which is here, you're getting pretty nice structure in the neural data. And then, of course, all the way out to 900 neurons. And these are of course, independently trained, but we can still see this nice representation of the feature time in these ways. And then what we can do is look across the consistency of the 2P versus neural pixels embeddings. And I think for me, I was quite excited to see that, you know, with around 400 neurons, with a high enough dimensional model, you can get quite highly consistent embeddings up to, you know, 85%. Where it does seem to be like the latents that we're finding in calcium or neural pixels is really similar. Of course, when we do this multi-session training, we can find even higher consistency with even less neurons. So now down to 100 neurons, you can find nearly perfect consistency. I don't think I have the slide here, but what we did do is also check that we're still seeing differences, so we're not wiping this out. So we checked between V1 and other higher visual areas, and we can still see this nice differences between the visual cortex areas in these mice. Okay, so the next thing that we did is that we wanted to look for this this kind of interpretability or this performance with decoding again. And so what we do is we build like a pseudo mouse decoding framework. So we take neurons from different mice. We build again this triplet, we build our embedding space, and then we add a simple KNN decoder. And so there's a few tasks that we did, so I'll explain these to you as we go. So the first one was just scene classification. So we annotated the movie and said when there was big scene transitions, because of course individual frames are highly correlated. And what you can see here is that especially in our jointly trained model, we have extremely good scene classification with a few hundred neurons of decoding off of this embedding space where they're jointly trained. This is for a singly trained modality on neuropixels data. And then here are 2 jointly trained, but simpler standard baseline decoding models in this way. We can also do this with individual frames. Even though they're more highly correlated, we still see a very similar trend in the data. And importantly, I think this was kind of, I thought was quite nice, is that even if you look at the exact true frame versus predicted, of course, there's jittering around the unity line. There's a few outliers here. It's not perfect, but it does a pretty good job of capturing what is the exact true frame versus the predicted frame. And we can also measure this by a mean error. So what is the actual frame difference number given the number of neurons? And it gets extremely low given neurons in the neural data. So to give you a sense of what this looks like, here I'll just show you a movie. On the left is just the embedding space that was trained with 2 photon and neuropixels data. Here's the true frame on top and the predicted frame on the bottom in this way. And I haven't seen the end of this movie, so if you guys know what happens, please don't tell me. I need to look at it another time. Just joking. But jokes aside, I think what's quite nice in the space is, one, you can investigate the dynamics. So I didn't talk about this much, but of course you can compute the flow in this embedding space and see where it is, where it's decoding these frames. And although it's a little bit jittery, and if you've watched it hundreds of times like me, you'll see that it's actually almost slightly ahead of the frame. So it's almost like the visual cortex is slightly predicting. Of course, the mice have seen the movie several times before that we're doing this, but this is, of course, held out cross-validated data that wasn't used for training the model, certainly. Okay, so, oh yes, I do have it, sorry. So we also examined then what the decoding was across different visual cortex areas, so including these more PPC-like areas and these higher visual areas, which I think is a field we still don't fully understand what these areas are doing in mice. And so here what we can see is that maybe a little surprisingly, but V1 has the highest accuracy of decodability compared to these other hyovizual areas, with the PPC-like area being the lowest decodability in the same task setting of predicting the exact frame. We also compared across different layers, so we can look at both in the neuropixels way and the calcium of where these neurons were laying, and we see that 2, 3, and 5 are more equivalent than 4, which is a bit interesting because, of course, 4 is the input layer, so this must suggest some prediction or recurrency in this data. Okay, but lastly, just one other kind of application for you is that you can just use this on behavioral data too. So if you take your deep lab cut key points, here's an hour of a mouse moving around in a freely running cage. What you can do then is very simply do action recognition on top of these spaces, right? It's a clustering algorithm at the end of the day, and find what these mice are doing. So maybe unsurprisingly, this big pink or purple loop around the space is when the animal is spending most of its time on the running wheel, but you can also do this for any types of data. So here is a case study from Sturman et al., where they have this nice elevated plus maze, which is a very classic test in neuroscience behavioral assays, especially for preclinical applications. And what we could show is that with our algorithm, even very simple things like head dips, we can recapitulate the ground truth much better than systems like EthoVision. And importantly, compared to commercial solutions like EthoVision, we see a much better match to the manual scoring. So it's not overestimating other parameters in the model. So with that, I hope I've convinced you that Zebra might be an interesting method on this era of big data and neuroscience to combine behavior and neural data across animals and mice. It produces highly consistent embeddings. It can be used with discrete, say, features or actions of the animal, or these continuous labels. We can generate topologically meaningful decoding embedders. Unsurprisingly, we find this low dimensional representation of space in CA1. I didn't go through all the data on the monkey today for time, but we can find this nice limb state representations. We can decode movies from V1, and it's truly data agnostic. So it can be used for behavioral neuroscience or any other time series data that you might be interested in. As I mentioned, all the paper is zebra.ai, or the preprint is zebra.ai, and very happy to take your questions. But first, let me just thank my team, especially Stefan and Jin, who I shouted out at the beginning, but of course, my whole lab and the funders for supporting our work. And thank you guys for your time. And again, I'm really sorry to not be there to get to interact with you more during this event. So thank you very much.
Okay, first, thanks for being on time. And we will try to take questions. Please have them concise because she can't hear you and I need to translate it. So not very lengthy, if you will. Any questions? Yeah, no way. What causes the shuffle data to collapse?
Yeah, great. No, great question. So basically in contrastive learning, because it's actually such a hard space to optimize, if you don't build out positive and negative pairs that are meaningful in the data, it's just really difficult to optimize, basically. So this is a known feature. Like There's another sort of school of thought which finds this as a bug, right? That you should always be able to find an optimization space. But I think particularly for our setting, I don't see it as that way. I don't know, you can see Jan LeCun tweeting about these things recently, but this is a known problem or feature, depending on how you want to look at it in contrastive learning, because of the complexity of the optimization space. Shortly, but I mean, I'm happy to expand more, but that's kind of the short answer.
More questions? Yes, hi.
What is the role of the latent embedding? Just take the pure data and pass it through this network. We have both cognition forms.
Yes, yeah. So I didn't show that, but yeah, definitely. So the other decoders that we used are like simple CNNs. So we use the exact same backbone as Zebra. So by not first pre-shaping this latent space, if you will, the decoding performance is much worse, and also the consistency that we find in the embedding spaces that are generated. So we do think it's a useful step, right? If anything, it's a dimensionality reduction step. You can think of it that way. I think it's potentially interesting for hypothesis-driven discovery of spaces, but it does seem to matter than versus just passing it through like an RNN or a transformer. I didn't show it today, but we've also benchmarked it against auto LFADs, and it also does better both in consistency and decoding than LFADs as well. So, which would be an example of an RNN variational autoencoder.
Okay. Thank you, Mackenzie. Let's thank Mackenzie again. Hi.
Thank you.